To the Chris Khan Academy, the YouTube channel, which brings you about to mathematics and the sciences. Now I'm coming up on biology. Dear students who do biology, I'm coming up. It's your period to learn from this YouTube channel. It's a great time to do exercises about biology, different questions. Here I'm coming up with a good package for you. The questions and the answers very correctly. I do research on you and then I help you to do this. This will help you to pass your exams. Come up with this. Here, it's about the many questions as much as possible, but with their respective answers. And then here, be the first or be the one to learn from this video such that you can pass your exams. If a teacher brings this question, make sure that you will do your best. Here, I'm going to start from question number 1. Here. So, is there any wrong statements about the prepeptides among the following? Answer by yes or no. Here, we have A, B, C, D. If there is a wrong statement about the prepeptides among the following, you can say yes or no. But for me, the real answer is no. Because in the prepeptides, the sequences of amino acid is determined by instructions on the cell's DNA. And they have an amino group at one end of the polymer. They have a carboxyl group on the one end of the polymer. They all called protein if they fold into a specific shape. There is no wrong statements. Question number 2, when a DNA stands with the sequence, when a DNA strand with the sequence 3 end to 5 end is used as template on which a daughter DNA sequence is formed, the daughter strand has the sequence. The real answer is number C. If they have 3 dash and 5 dash as the ends, and then their daughter DNA sequence to be formed, it must start from 5 dash to 3 dash, but starts with C because cytosine paired with guanine. Number 3, it's all about vocabularies. For each of the following definitions, give the appropriate terms and spell it correctly. So terms are single words unless indicated otherwise. So here we have Roman 1. If we say that the method of DNA replication in which each strand of double helical molecule serves as a template against which complementary new strand is synthesized, it's what we call semi-conservative. And then if you say a genetic locus that serves as recognition sites for RNA polymerase attachments, it's what you call promoter. And then if you say a group of 3 nucleic acid or nucleotides in messenger RNA that specifies an amino acid, it is what we call a codon. And then to be called a RNA primer, it's a short RNA sequence onto which DNA polymerase 3 adds a deoxyribonucleotides during bacterial DNA replication. And then the tertiary structure, it's all about called the complex process by which prepeptide chains attain a stable 3-dimensional structure through short-range chemical interaction between a near amino acid and long-range interaction between amino acids in different parts of the molecule. And then an alpha helix, it's all about called a spirosecondary structure in part of many peptide chains, and then consisting the secondary level of organization. The process whereby RNA is synthesized from DNA templates, it's what we call transcription. And the lagging strand, it's all about a single-stranded pieces of DNA produced by a discontinuous replication of double-stranded DNA. And then introns, it's likely to be called the regions within a eukaryotic primary transcript that are removed during the process of messenger RNA. And the number 10 was a definition of hybrid DNA. A hybrid DNA is a DNA segment generated during recombination by same pairing between complementary and single strand from different parental duplex molecules. So Here we have question number 4, which is completing the following sentences. If we said that the sequence necessary for transcription, it is what we call a promoter, and then dash dash single or a signal the end of transcription, it is what we call terminator, and then the name of a newly synthesized eukaryotic RNA, it is a pre-messenger RNA, and it must be modified before it lives in the nucleus. Dash dash general encode protein that are used in metabolism or biosynthesis, what we call structural. Lac operion is inactivated when lactose molecules are absent in the cell. Yeah, the general. So genes in prokaryotes tends to be grouped together in operon, and then the bond between the phosphate group and the ribosome sugar group in RNA is called the phosphodiester linkage. So the subunits of RNase polymerase whole enzyme, which is responsible for promoter recognition, is what we call a sigma. Splicing is the process that removes introns and converts exons. Helicase enzyme unwinds the DNA molecules for replication. So semi-conservative DNA replication was demonstrated by M. Meselson and Y.F. Stahe. The secondary structure of protein is determined by heterogen bonds or the separate bond. So nucleic acid are a polymerase composed of nucleotides, a monomer, as monomers. So RNA in the ribonucleic acid, DNA is ribonucleic acid. So how does DNA replication differ between eukaryotes and prokaryotes? For both in eukaryotes and prokaryotes, the DNA replication in eukaryotes is linear replication, whereas for prokaryotes it is actual replication. So the DNA replication in eukaryotes, the two linear DNA molecules are produced, whereas prokaryotes, the circular multiplication molecules. So several multiplication origin at several sites on the eukaryotic DNA replication. Prokaryotic DNA replication have a single origin of replication. So here the human genome comprises all about this. It's protein coding genes, gene encoding messenger RNA, sequences that do not encoded genes or gene for ribosomal RNA and the transfer RNA. Here we have the RNA polymerase for Escherichia coli is a multi-subunit protein composed of it's a beta, no, it's a sigma, yes. Eliho independent transcription terminator sequences prokaryotic DNA has a GC rich region followed by a string of the adenine. Here we have a question considering the following region of replication that found in a chromosome. The sequence of region 1 is shown below, and then you have which region within region 1 which strand will be the template lagging strand synthesizing. Is this the top or the bottom? It's have to be the top. Then here our mount, the question is about this structure. Here there's number 10. I will come up with the real answer of this question number 10. And then number 11, we have which of the following an example of missense mutation. This is UGC to UCC. What's called to be the structure is what we call anticodon. This is the question number 12 with their respective answers. The answers are those on which within yellow. Number 13, I have a real answer with them. I will show you the video for this. Which of the following messenger RNA molecules would form the most stable stem loop structure? It is number B. As it is, these are questions you have to learn from this, and then you do your exam excellently. Methionine is coded for by one codon only. Histamine or histone is a DNA in package into chromatin with the help of the protein called histone. This is all about Okazaki fragments are the newly synthesized strands of DNA produced during the replication of the lagging template strand. What you call stop codon. It cannot play a role in replication. Yeah. The human genome comprises protein coding genes, gene coding messenger RNA sequence of that do not encode genes, and so on, as it is outlined here. These are questions that can help you to pass your exam in biology. Those are questions which don't have which do not have answers. I'm there for it. I'm come up with a new video which can help you to learn from this, and then you do your best. Pendrome. Yeah, this was all about the questions, 48 questions, that this can help you to pass your exam excellently without any challenge. Watch this video clearly and story by story, and then you will do your best. Thank you very much. It's a package for students.